Hello, my name is Johar, and we are going to discuss about enzymes in this session. So I had divided this session into two parts. So first part is this, and then there will be later another part. First of all, the objective of the session is to define various kind of terminologies like enzymes, substrate binding site, allosteric site, holoenzyme, apoenzyme, and so on. There are many terminologies. We must get acquainted with these terminologies at the end of the session. And we have to understand how we can classify the enzyme, what are the different categories in which enzymes has been kept by the scientific community. Another thing we must understand at the end of the session, how enzyme interact with this substrate and make the product, what are the mechanisms. At the same time, we will try to understand the major properties of the enzymes and what are the factors that affect the activity of these enzymes. So let's begin. First of all, the definition, the enzymes. Enzymes are, we can say, biocatalysts. Majority of them are protein in our body, with a few exceptions only. They accelerate the rate of chemical reaction by lowering activation energy. So they do not change the equilibrium. It means that if a reaction cannot happen, so they cannot let it happen. But it can also only increase the rate of reaction. If a reaction is feasible, then only enzyme can increase the rate of reaction. So they are very specific. One enzyme can only act for certain kind of reactions. It cannot be enzymes for all kind of reactions in the body. So one point is very important here. Majority of the people confuse that the enzyme have no effects on equilibrium. It means that suppose if A combines with B and makes C and D, so these are the substrate and these are the products. So this equation, this chemical reaction is feasible in nature. It means A can combine with B and make a C and D. So these are substrate and these are products. However, let's take another example. A combines with B and makes E. This is another situation where A and B combines and make E. Let's another situation. A combines with B, but we are trying to make, let's suppose we are trying to make another, like for example F, and it is not possible. In nature, it cannot happen. So an enzyme cannot make it happen also. Enzyme can act on this. Let's, for example, what exactly enzyme do is, let's for example, while combining A and B and making C and D in a reaction, it takes approximately 2 days to make one product, like one A and one B combined to make one C and one D in 2 days. So what exactly enzyme do is decrease this time. That means increase the rate of reaction, change the substrate into products at a faster rate. That's all. This is only the enzyme is doing. It cannot make a reaction happen. Like for example, A and B cannot make F, so enzyme can also cannot do anything here. So any reaction that is thermodynamically not feasible, like I'm writing here, let me write here so that there won't be any confusion in future. Any reaction that is thermodynamically not feasible, not possible or feasible, then enzymes has no role in it. It means that enzymes has a role only in those reactions when it is thermodynamically possible. it can happen. The only thing enzymes do is accelerate the rate of chemical reaction by lowering the activation energy, that's all. The enzyme, without enzyme, like for example, another example I'm giving you, like for example, if we do not have an enzyme, then a particular reaction can take several years, like for example 10 years to take place, but with the enzyme it can take in fraction of seconds. So this is the only doing by the enzymes. Now the next is the substrate. What is the substrate? Substrate is a substance which the enzyme acts to form the product. But as we discussed in the definition of enzyme, anything that cannot be made by those substrate, so in that condition enzyme cannot act. I mean, enzyme cannot do it, let it happen. The enzyme binds to its substrate to form an enzyme substrate complex. The binding of substrate with enzyme takes place at substrate binding site or active site. So the substrate binding site or active site, or we can also call it catalytic site or isoesteric site. Isoesteric site. It is a small region on the surface of the enzyme. It has a specific chemical groups that bind a specific substrate. The substrate binding site is the enzyme active site or catalytic site. For example, here in this situation, like this is an enzyme, this is a substrate, here is the binding site. The substrate cannot bind anywhere else, or it cannot bind anywhere wherever the substrate wants, or it can find it. It can bind only at a certain location, and that specific location or that specific site is known as active site or catalytic site or isoesteric site or substrate binding site. All are same. Now the another definition is the alloesteric site. Any site other than, allo means other than substrate binding site is known as alloesteric site. For example, here if you see, this is the substrate binding site or the active site, and this is the alloesteric site, because here substrate is not binding, or substrate does not bind here in that particular reaction or in that particular enzyme. So significance of alloesteric site is that there are many things that binds the enzyme at alloesteric site or other sites other than active sites and modulate its activity, increase or might be decreased. There are other terms like holoenzyme, apoenzyme and coenzyme. Enzymes are mainly protein in nature, as we have told earlier, as we had discussed earlier as well. Some of them are simple proteins that do not require additional chemical groups to act, like pepsin, trypsin. There are many enzymes which does not require any accessory group. However, there are other enzymes that needs another accessory groups, and that kind of enzymes are known as conjugated enzymes. Conjugated enzymes are formed from a protein part called apoenzyme and non-protein part called coenzyme. So there are two things. One is the protein part that is known as apoenzyme, and then one is the non-protein part that is known as coenzyme, and the entire enzyme is known as holoenzyme. So if you see here, this is the apoenzyme, this is the protein part, and this is the cofactor. Both will make the holoenzyme. Without this, enzyme will not work. So this is important. It must bind here, and then there will be an active site, substrate will bind here, and then it will make a product. So holoenzyme, certain enzymes can only act in a holoenzyme form. That means, however, certain enzymes can act only in the apoenzyme form, like there is no need for any other component, or they are not conjugate enzyme. But certain enzymes need another component like coenzyme or cofactors. So these are known as holoenzyme. Now how the enzyme actually work? The formation of enzyme substrate complex is one of the most important steps. The first step in an enzyme catalyzed reaction is binding of substrate to the active site of the enzyme to form the enzyme substrate complex. This complex is then converted to a product, and enzyme is regenerated again, ready for the second reaction. So what it means that enzyme makes a complex with a substrate, and then after certain chemical modification there will be a formation of product. But in this situation, one thing is very important to remember, that enzyme does not get utilized in the reaction. I mean, enzyme remains as enzyme after the reaction also. So enzyme does not get utilized. Only the substrate will be converted into products. Now enzyme here is the E is the enzyme, S is the substrate. It will bind, and they will make a enzyme substrate complex, we call as ES complex. This making of complex is a reversible reaction that can happen in both the sides. Now enzyme substrate complex can go back to make enzyme and substrate also, like for example this is however, when the enzyme substrate complex can make a product, here enzyme plus product, so enzyme will remain the same at the end of the reaction, only the substrate will be converted into product, and that production or that conversion is very enhanced, or we can say that the timing or the duration is very minimized. That is the enzyme is doing these things by lowering the activation energy, so that rate of reaction can be increased. So by pictorial representation we can say this is the enzyme and this is the substrate binding site. Now what happens, the substrate will bind with the enzyme. This will make enzyme substrate complex. The substrate is this, enzyme is this, and then it will make a products and enzymes. So how we name the enzymes? There must be some mechanisms to name the enzymes. Usually at the end of the enzyme there is a word is. Most enzymes are named by adding suffix. Suffix means, suppose there is a word hydro. So if we add lase after that, it is known as suffix, and if we add like pan hydrolase, so pan is become a prefix and this will become a suffix. So anything that is add after that particular term, and usually it is added in case of enzyme is ase. So usually majority of the enzymes, with a few exceptions, of course with a few exceptions, but majority of the enzymes are having ase at the end of their names. Some notable exceptions are there, like for example pepsin, trypsin. And as we go ahead we will discuss about, there are 6 major classes of enzymes. These are categories of enzymes. They have been divided depending upon their activity, what kind of reaction they catalyze. The first of them is the oxidoreductases. It basically catalyzes the oxidation reduction reaction. Here lactate is converted into pyruvate. This is a oxidized condition and this is a reduced form of pyruvate. So the enzyme is lactate dehydrogenase. So lactate dehydrogenase comes under the category of oxidoreductases. Now the next enzyme is transferases. Transferases is usually transfer one kind of group from one substrate, one molecule or one compound to another substrate or another compound. Like for example, here if you see the methyl group, that is the CH2 group, is transferred from serine to tetrahydrofolate. So serine become glycine. As we know that there are 2 carbons in the serine, it's an amino acid, and in the glycine only 1 carbon is present. There are 2 carbons in the glycine and 3 carbon in the serine. So while transferring 1 carbon from serine to the tetrahydrofolate, it will become glycine losing 1 carbon, and this 1 carbon is transferred onto tetrahydrofolate. And tetrahydrofolate is basically act as a methyl donor while getting these things. So this reaction is catalyzed by serine hydroxymethyl transferase, and this enzyme comes under the category of transferases. There are another very classical example of transferases is the transaminases. There are many transaminases in our body. However, the amino transferases is very important. It converted the amine group from one to another. Like for example, in the aspartate, it takes the amine group to the alpha ketoglutarate, and while transferring of amine group from aspartate to alpha ketoglutarate, it becomes glutamate, and aspartate becomes oxaloacetate. So the reaction is catalyzed by the transaminases or aminotransferase. This aminotransferase comes under the category of transferases. The next group is the hydrolases. In this, the cleavage of substrate makes two products by the addition of water. It is important by the addition of water, because cleavage can happen without addition of water. That is another set of enzyme. Here the urease is catalyzing the reaction. Urea is converted into ammonia and carbon dioxide by the addition of water, and urease comes under the hydrolases. There are another enzyme that is lyases. Here they used to cleave the compound, but they do not add water. Here example, pyruvate is converted into acetaldehyde. Here one carbon dioxide is released, one carbon is released. However, there is no addition of water. So this kind of cleavage without addition of water comes under the lyases, and the enzyme catalyzes pyruvate decarboxylase. So this enzyme comes under this category. Now the isomerases, they only change the orientation. The chemical formula will be the same, but only that there will be change in the structure of the molecule. So for example, methyl malonyl coenzyme A mutase is one of the isomerases, and it comes in this category that catalyzes the isomerization of methyl malonyl coenzyme A to succinyl coenzyme A. We will learn this will be utilized in the tricarboxylic acid cycle, and this will be a byproduct in the breakdown of the oil chain fatty acids at the last step. Ligases catalyze formation of bonds between carbon and oxygen, sulfur, nitrogen, coupled to hydrolysis of high energy phosphates such as. So basically in this condition there will be a joining of two bonds, like carbon is joining this. So this is formation of new bonds. So two substrate become one, make one product only. Here the enzyme is the pyruvate carboxylase. This kind of reaction needs an energy. So ATP is providing the energy, and this enzyme pyruvate carboxylase is converting pyruvate into oxaloacetate. Pyruvate is a 3-carbon and oxaloacetate is a 4-carbon molecule, and this enzyme comes under the category of ligases. So far we have discussed about the different categories. So there are 6 categories, and now we are going to discuss about what are the different factors that affects the enzyme activity, because it is important to know how we can increase the activity of the enzyme if we need more product, or if we do not need more product. In fact, we want to decrease the product concentration, then how we can reduce the activity of enzyme, or how can we can create the impedance while the enzyme is acting. So there are 3 important components that affect the enzyme activity, while if we do not add any poison or any inhibitors in the reaction. One is the substrate concentration, the other one is the temperature and pH. In the next session we will discuss about the addition of the external components like poison and enzyme inhibitors as well. But here we are talking about absolutely normal situations where inside the body things are going on or reaction is going on, and it will be only affected by either the temperature, the pH of the cell or intercellular compound location or components, or the substrate concentration where the reaction is taking place. So one important thing here to mention is that if you want to study any of the component or the factors, how it is affecting the enzyme activity, then you have to keep the other two. Like for example, if I want to study the substrate concentration, what is the effect of substrate concentration on enzyme activity, then we have to keep these two, temperature and pH remains constant. We shall not change the any temperature or pH in the in vitro condition, like in the laboratory if you are doing this. So how substrate concentration affects the enzyme activity? Like here in the graph it is mentioned, if we increase the substrate concentration, enzyme activity will be increased. However, after some time it will reach a plateau. So even after this concentration, like for example this concentration, even after this concentration we increase the concentration of the substrate, but there won't be any change in the speed of the reaction or the enzyme activity, because all enzymes get saturated at this point. Okay, so increasing the substrate concentration while all other conditions are kept constant will increase the initial velocity V to a maximum velocity. It is a hyperbolic curve. So ultimately it will reach to a maximum velocity. This at this concentration, this is the maximum velocity and this is the concentration. At this concentration the activity of the enzyme is maximum. So here we can calculate the velocity of the reaction. The initial velocity is the velocity measured at the beginning of the reaction. And there is one more important thing here. If you see, this is the half of the maximum velocity. Okay, this is the half and this is the half. So half of the maximum velocity reached at certain concentration of substrate. Okay, this substrate concentration is known as Km. Okay, this is also known as Michaelis constant. So there are three things here that needs attention. First is the Vmax, that is the maximum velocity that is reached at certain concentration. Even after that concentration, even if we increase the concentration beyond that point, there is no change in the velocity or the speed of the reaction. The another thing is that we have to remember there is a half of the maximum velocity, that is Vmax divided by 2. Half of the maximum velocity, and this can be reached at certain concentration of substrate. That particular concentration of substrate is known as Km. It is a certain kind of constant for some reaction additive. And the third important thing we have to remember that this is a hyperbolic curve. Now this is a Michaelis-Menten reaction. Here this is, as we discussed, Km. Km is the substrate concentration at which rate of the reaction of an enzyme is half of its maximum. And Km is a specific for each enzyme. So it is an enzyme specific constant, and it is the substrate concentration at which the reaction has its half velocity of its maximum. So if we want to know the rate of reaction in a particular reaction, that is the V0 at any particular time, like 0 is basically initial velocity, but if we want to measure at any particular time, then at that particular time we have to know the substrate concentration. And there are two constants, Vmax will not change and Km will not change. So these are two constants, and we just know, if we know the substrate concentration keeping the temperature and pH remains same, then we can know the velocity at or speed of the reaction at any particular period of time in the reaction. So now what is the significance of this Km or the Michaelis constant? The significance is that in a high affinity of the enzyme, if the enzyme is very much affinity with the substrate, then the Km or the Vmax, half of the Vmax will be reached at a very low concentration. So half of the Vmax will be reached at a very low concentration of substrate. It means lower the Km, higher the affinity. Lower the Km, higher the affinity, of enzymes towards its substrate. Now we will learn this significance of Km in glycolysis. There are many other significances also, but in the glycolysis there are first reaction is catalyzed by hexokinase, but there is another enzyme that we know as glucokinase. although both will catalyze the same reaction, that is, the glucose will be converted into glucose 6-phosphate with the help of ATP. However, the hexokinase and glucokinase has different affinities with their substrate like glucose. So we will learn about the significance of Km when we talk about glycolysis. Low Km means there is a low affinity means there is a high Km. So now the temperature, if we increase the temperature, the rate of the reaction will increase, but up to a certain extent. After that, even if we increase the temperature, there will be decrease in the rate of reaction. So at certain temperature, there will be maximum rate of reaction, and that temperature is known as optimum temperature. In humans, it is 37 degrees centigrade. But in other organisms, it changes with different kind of organisms. Like for example, there is an bacteria we call as Thermus aquaticus. We used to name it abbreviation TAC polymerase. They have an enzyme like DNA polymerase that can act at 95 degrees centigrade. So we use this enzyme, this bacterial enzyme, in PCR, polymerase chain reaction. And we know that there are many things we do. It's a diagnostic test or screening test for certain things. So TAC polymerase, Thermus aquaticus, it's bacteria. We use its polymerase, and the optimum temperature for this enzyme DNA polymerase is 95. However, in humans it is 37 degrees Celsius for majority of the enzymes. So that is known as, that 37 degrees Celsius where the enzyme activity is maximum is known as optimum temperature. Now the pH. For every enzyme there is some optimum pH at which the activity of the enzyme is maximum, activity of the enzyme is maximum. So like for example, the pH of the human blood is 7.4. So in case certain enzyme is present in the blood, usually the optimum pH is around 7.4 for that enzyme. If like for example, some enzymes are working mainly in the stomach, like pepsin, the pH of the stomach is very low, so their optimum pH is 2, because if they enter into the small intestine, they gets deactivated because of the high pH in the small intestine. So that is all about the first part.